This is the BioCentury Show.
Brought to you by BioCentury Grand Rounds. Join BioCentury and Regional Host Chair Allen Institute this June in Seattle for an R&D forum at the interface of industry and academia.
Hello and welcome to the BioCentury Show. I am Simone Fishburn, Editor-in-Chief at BioCentury. So delighted to be joined today by Chris Hollowood, CEO of Syncona Investment Management Limited, and I would say one of the leading voices also in the UK on UK biotech. Chris became CEO of Syncona in 2023, having been at the firm for over a decade, previously as Chief Investment Officer. His background includes several years in the investment world. Before that, a degree and PhD in chemistry from Cambridge. For those of you in the know, meaning he's yes and Natsky. So we'll see how that plays out during this conversation. You're going to have a backpack or something. I don't know. All right. Chris, let's start at a pretty, I don't know whether to call this a high level or a deep one, right? I want to talk about rational target discovery and where the industry's going. So there's just a lot of talk about causal biology being the North Star. But you've got a sort of slightly different take on this, which you discussed last year at our Grand Rounds conference. And I'm going to quote you here, okay? You said, if the underlying biology is a premise of the experiment, then it cannot be used as evidence of the causality. See how erudite? Okay. Oh. Can you expand on that for the audience and talk about what your concern is here?
Yeah, well, so firstly, thanks for the invitation to speak today. And I sounded very eloquent a year ago, so hopefully I can repeat it here. But the fundamental issue I see is part of causality. One of the tests of causality is plausibility. And if you look at the Bradford Hill criteria, that's in there. Actually, if you read Bradford Hill, he had concerns about using plausibility as part of that causality proof. And the issue, I think, historically in biology has been you start with a great professor that has studied a piece of biology for 10, 20 years, and they come to you and go, this is a great piece of biology for this disease. And then they run a set of experiments to prove causality. And those set of experiments give you the set of results, and they go, the results all line up. And because the biology is plausible, it must be causal. And that's the issue. You can't have the hypothesis be the proof of causality at the end. And I know we're going to get to it, but I see the industry is actually on the cusp of solving this problem. I see a very exciting future for the industry because I think with the tools we have nowadays, we can get around this issue and get much more powerful causality arguments into our experiments than we could before.
So I want to just sort of iterate through that for a minute. So let's say somebody finds a gene association with a particular disease, and then they find a pathway that gene is involved in that's plausible for the disease. Is that still circular?
No, that's, as we know, that's the pinnacle. Because
that's the pinnacle.
Because genetic evidence, as we know, is a great predictor of a biological target. But the point being, you didn't start saying it's that gene.
Right.
You looked for a gene that correlated, and then you checked its biology, and its biology was plausible. That's the right way around. So that works beautifully.
Right. So you've talked a little bit about the importance of an unbiased step. And so that genetic step would be the unbiased step, I assume. So maybe you're going to expand now, on what tools the industry's got, why you think this could be, I don't know if you want to call it a pivot or sort of like the cusp of a new era. What is exciting?
Yeah, so I think it's not a pivot, it's a natural progression. But when you look at the tools we have available to us, like CRISPR, like machine learning, where you can effectively go into a biological system and knock out each gene individually in an experiment, and therefore you're not entering that experiment with a biological hypothesis of it's this pathway or that pathway, and then look at the outcome as to which are the most strongly correlated in that experiment to the outcome you wanted, and then check plausibility, then the plausibility can be used as proof of causality. And because that is a holistic experiment, I mean, Bradford Hill talked about it, that, where you see associations, you can't rule them out on the lack of plausibility because you are at the frontier of what is known on that day.
Right.
And we've been limited in the past. And now, because we can do these holistic experiments, not only is the experimental design more powerful, it covers a lot more biological ground, and you're more likely to find the best target, not just a target that might work. And it's those two elements of it that I think are going to hugely de-risk novel target discovery and therefore drug development going forward.
So I want to take a tiny tangent here. So this is a personal association of mine, somebody that I know who had absolutely no symptoms, and they found a homozygous genetic mutation. It's actually in the eye, right? No symptoms at all. And they found it when they were screening for something else, right? So this individual was told, okay, you have this genetic abnormality, but she had no symptoms. And so even though there's a very strong association, all of the people with the disease have this genetic abnormality. There's no information about the number of people with this abnormality who don't have the disease. So this is why I sort of went back to that genetic thing in the first place. For me, it's not necessarily circular, but there's still this tremendous amount of information that we don't have. You can have a strong correlation, but you don't know if there's a protective factor or something else. How do you think about that and how biology should be solving it?
Yeah, so I think that's a very pertinent question. I think in your example, what you cited as the correlation isn't as strong as you thought.
Okay,
so, yeah,
right. So.
Because the penetrance of that predicted genetic mutation has been misunderstood, because the people that are presented at clinic are the people with the disease.
Exactly.
And not look holistically across the population as to whether you have that mutation and the disease as well. So that goes to strength the correlation. And then if you did see those patients separate out into ones that got the disease and then a clear set that didn't, then yes, you would look for a second marker, some of the things you described there, as to why is that happening, and maybe discover, what is mitigating, and maybe that will give you an idea to treat the disease.
So I think you're right. I totally agree with that. And that, it's the patients who present, right, that the data's from. But what is the solution? Is it the sort of whole scale? I was going to get to this later, but we're down this road now. Is it the kind of wholesale genetic kind of screening of population so that you have just a much fuller data set? How do we go about that? And do people want to all be genetically profiled and screened?
I think there's two questions in there. But look, I think from an experimental design point, you need to make sure that you have done a holistic experiment and it's unbiased. And that gives you the greatest confidence. I facetiously think about, biological target discovery historically as that person feeling the backside of an elephant and trying to figure out what it is. We can move away from that. We can look at the whole elephant now. And that, I think, is much more powerful. And the second question is, do people want to know? Do people want to enter into those kind of studies? I think that's a societal question.
Right. So just, I'll come back to targets in a minute, but I do want to talk about the way you are approaching this within your companies and how that changes the risk paradigm for you and where it leads at a high level. I mean, is it a tangible difference in the outcomes with this new sort of way of looking at it?
Well, so right now we don't know. So we're venture capitalists that have a thesis, and we're basing a lot of our investments on the back of it. I think all the evidence does point to it. So, AstraZeneca put a paper out, I think it was the mid-2010s, that talked about phenotypic screening versus target-based discovery.
Right. Yeah.
And the greater correlation to drug approvals. And I know there's been a lot of opinion on that paper, but I think that is a very interesting set of data that supports the unbiased nature of what we're doing. So there's one evidence set there. And then the holistic piece that we're adding in, it's hard to argue against that piece. And then the flaw in the holistic piece is what is the experiment you're doing, and does it replicate disease? So it's one thing doing a holistic experiment across an in vitro assay versus a holistic experiment in a human being. They are completely different standards of proof. And we know that from all the other work we've done. So we do look for holistic, we do look for kind of plausible biology, not as the hypothesis, but as the proof. And where we can do it in a human, that's the absolute pinnacle.
Right, right. No, it's extremely interesting. I want to go back to the point that you made about targets, right? Because this is so often the holy grail, and we just published, our analysis of abstracts at AACR, where we identified 176 potential new cancer targets, right, that weren't in our database and hadn't been previously associated. But you and I know targets alone do not a first-in-class make, right? So
No.
modality
Right.
matters. Okay.
Yes.
Talk a little bit about that. And I know, you pursue that through your portfolio companies, Yellowstone, PureSpring. Talk a little bit about your approach to modality plus target, that solution.
Yeah. So I think we've had a big paradigm shift in the research tools that allow us to think about target discovery differently. What we saw over the last 12 years is a huge addition to the set of drug formats we've got. You go back 20 years, you kind of had an antibody and a small molecule, and if it was a target within a cell, you kind of stuck with a small molecule. As a chemist, I didn't mind that, but that wasn't a great tool set to have. And now you've got RNA, you've got CRISPR, you've got gene therapy, you've got cell therapy, you've got all of these different modalities and engineered versions thereof. So you've got this really rich set of things to go at targets with, but it also presents jeopardy because now I don't think it's sufficient to be first in class for two reasons. One is if you were first in class before, no one could come around the side of you because if you had a small molecule, that was the only way of getting at it. Now, if you haven't got your modality to match for the target biology you're going after, are you hitting it precisely enough and hard enough? And the disease context, does it mean that that therapy is in a format that the patient with that disease wants to take? Then, given how IP is narrowed, I think you're in real danger of getting overtaken. And we've seen the pace of development now, particularly you've seen China really up the pace of development. So you're not safe unless you get all of those things right. So we have a company in our portfolio, which I'll use as an example. This company's called PureSpring. We've done a lot of investments in gene therapy and been pretty successful at that, particularly in the eye. And really what you're looking for is diseases in terminally differentiated cells. So we thought, let's look at the kidney. There's a huge number of genetic disorders there. It's a terminally differentiated cell. And then we have a view that mechanical delivery can do so much more than biological engineering. So whilst most people out there would have thought, I'm going to engineer an AAV to make it renal trophic, we thought that was a very high bar and a very long road. And actually, we looked at what the surgeons do. And so surgeons are able to clamp the renal artery and infuse drug for 20 minutes downstream of that clamp and perfuse the kidney doing that. So we took a known surgical procedure and decided that that was the route to delivery. If you're doing that, you don't want to do it very often. And so that made gene therapy the modality. And in the kidney is a monogenic recessive disorder called Alport disease, which is a horrible disease. No disease-modifying therapies out there, none really in development. And it takes a genetic medicine solution, in my view, as monogenic recessive disorder, it takes a genetic medicine solution to address it. And so there, I think we've done an exquisite matching of modality, delivery, disease in order to get the right therapy into it. And gene therapy is not the answer to everything. Gene therapy is for some things like monogenic recessive disorders. And in other cases, it's bispecifics, in other cases, antibodies. But you've got to get that matching right, because if we'd gone at it with something different, then all we would have done was prove it's a good target and that you can get clinical traction in it, and then we would have got beaten.
We'll come back and talk more about cell and gene therapies in a minute. Before we go to break, I just wanted to sort of circle back, because what most people do with a gene therapy is they would, except with the eye, they would deliver it systemically and assume that you need a targeting agent to get it to the kidney, let's say, which is very difficult, obviously. So what you're saying is that it's almost like a local delivery there, effectively.
That is a strategic decision we took very early, and we haven't backed off, because if you look at the dosing for kind of a liver-based gene therapy and a muscle-based gene therapy, they're an order of magnitude different. And the reason they're an order of magnitude different is because about 10% of the gene therapy doesn't go to the liver. So there's no targeting going on there. What you're doing is cranking the dose to get enough through the liver to get to the muscle. And our view is that, firstly, that was difficult from a CMC perspective, but also you ran a lot of risk against therapeutic index. And why not just do a low-rent solution, inject it directly where it's needed? And that seems to work.
Great. We'll take a break. And as I said, we'll be back in a minute to continue the conversation.
This episode of the BioCentury Show is brought to you by the third BioCentury Grand Rounds in Seattle. Advancing drug development requires more than discovery. It requires the right partners. The third edition of BioCentury Grand Rounds US convenes venture capital, biopharma decision makers, and academic innovators in Seattle, June 3rd to 5th. This R&D-focused forum brings together leaders at the forefront of translational science to examine the breakthroughs, bottlenecks, and strategies shaping the future of drug and diagnostic development, and how to make early-stage R&D investable. Discover cutting-edge disease biology and platform technologies. Gain insights from emerging biotechs and academic pioneers. Schedule partnering meetings with VCs, pharma, and leading academics who can accelerate your path forward. Grand Rounds US is where rigorous science meets strategic capital, and where the right conversations move discovery toward development. Join us in Seattle and discover what's next in biopharma and who's driving it. Secure your spot. Register at biocenturygrandrounds.com.
We are back at the BioCentury Show, and I'm delighted still to be here with Chris Hollowood, CEO of Syncona. So, Chris, it has been actually quite a tough time for gene therapies. We talked a little bit about it, your investments there. You had some exits with Nightstar and Gyroscope. Those therapies didn't work out. You still are in that space with Beacon. And generally, I think people with gene therapies are facing a lot of challenges. Perhaps you can talk about those challenges and how you are evolving a strategy and what you think the field needs to do.
Yeah, so I think gene therapy is not a solution to everything. We never thought it was, and we never planned to take it to everything. What gene therapy does brilliantly is, in a disease terminally differentiated cell, change the phenotype. And there are a lot of genetic disorders in terminally differentiated cells. So then you get to the question of, how do I get to the relevant cells? And that's why the eye has been so successful, because I can inject in that. And then CNS has been harder, because although there's a lot of horrible genetic disease in CNS, getting to the right cells and the endpoints to show that it's working make that harder to attack. And then beyond that, you have to get to doses that are relatively high. And we were always nervous about that. So I would take gene therapy and I would break it down into, can I deliver a significant amount to a hopefully monogenic recessive, or at least a genetic disorder, locally? And if I can really drive therapeutic index. And if I can't, I'm running a lottery as to whether I can navigate between efficacy and toxicity in a modality that you can't reverse. It's not like you can titrate these patients up. And so I think a lot of people took gene therapy into those settings and have had issues as a result. So we never did. We had a very strong focus in the eye. That was the obvious place to go. And we had a great success in Nightstar. And I think if you look at the data in Nightstar, that was absolutely progressible to pivotal trial. And Biogen decided not to do that for strategic reasons. So I don't think it's a case of it fails as a therapy. And quite the opposite. I think the reason we're doing Beacon is because it absolutely works in our view, and we want to get that therapy to patients. And then in Gyroscope, similarly, Novartis took a strategic decision. And I do think that a complement modulator for dry AMD is a good gene therapy target, because I think those patients that are progressing over 10 years and are asymptomatic at the beginning would appreciate a modality where they don't have to go to clinic every month. So I think there's places you should play gene therapy. And I think there's places it absolutely shouldn't. And I think people went beyond that boundary and have struggled. I'll give you another example. So we have a program in Gaucher disease, and it's systemic. So we deliver it to the liver. Liver secretes GBA. And we took a view that in order to get the efficacy, we could do something very clever, which is you could engineer the protein. And engineering the protein meant that we had a GBA that was equivalent in activity, but 20-fold half-life. And it's the lowest dose systemic gene therapy in clinical trials anywhere in the world. And so I think if you're smart about gene therapy, you can use it in the right settings in the right way, but don't expect it to be something it's not.
And so that's Spur Therapeutics, the one that you were just describing. Right. Let's just get that name out there. So, now let's move to where you've had good news. Well, one of your portfolio companies, Autolus, if I'm pronouncing it right, Autolus, I'm never really sure, with approval for the CAR T cell therapy. Has that sort of ramped up your interest in cell therapies versus this? I'm going to ask another question about cell therapies for the minute, but let's just talk about Syncona and how you're looking at one modality versus the other.
Yeah, so I think Autolus has did a tremendous job, and I think Kristen Eaton, the CEO there, is a force of nature.
Absolutely.
And we're very, very proud that we got our product approved. It's not worked out as an investment yet for us, and I think that taught us something about cell therapy, which is these things work. And quite clearly, you look at the efficacy and the safety in particular with that drug, that is something very meaningful for patients. But you need to target it at large markets, because it's only a large market that can actually withstand the infrastructure cost and allow you the leverage of that to really make it into a pharmaceutical product. And so we do have, beyond Autolus, we do have two other cell therapies in the portfolio. One addressing end-stage liver disease, which is Resolution, has some really remarkable early-stage data there. And then Quell, which is looking at autoimmune conditions, again, big market. So we focused our portfolio down to those two opportunities because I think they work for cell therapies. And I think cell therapy at its best gets you to an efficacy envelope that no other modality can get to. But you need to go to places where that level of efficacy is needed, because if another modality can do something sufficiently good, people are going to use the other modality.
Right. Yeah, I mean, people quite often bundle cell and gene therapy. And for me, the dynamics of each one are really quite different, like cell therapies versus gene therapy.
I 100% agree with that. I think it's easy to bundle them together because they came online around the same time. But I think the diseases you go after, the development dynamics, the commercial dynamics are all very different, actually, between cell therapy and gene therapy. And we certainly think about them very differently.
So I will come back to this in a minute because I want to sort of have some questions. Obviously, one of the big things with cell therapies is the role of China and how they're driving that. But before I go there, you talked about your strategy. I'm very, very interested in how Syncona is moving forward. You announced a pivot last year. You've got a clear message that you guys are open for business. I know you're trading below your net asset value, which I'm sure is not where you want to be, and you have a strategy for closing that gap. Perhaps you can talk about your strategy moving forward.
Yeah, so during the kind of nuclear winter that we've just been through in the bear market,
we've all been through, yeah.
We've gone through it. We worked incredibly hard to push our portfolio to late stage. So 3 years ago when I took over, the portfolio was about 25% clinical stage. It's now over 85% clinical stage, and that portfolio will fuel the growth. The Beacons of the world will fuel the growth of Syncona and provide the proceeds both to allow us to close the discount gap, but also to reinvest in new opportunities. And on the sort of being a London listed entity, the benefit of that is it gives us permanent capital, which I think in Europe is a huge differentiator where there is no depth of capital at any one stage. And so one investor that can connect the dots from university spin out all the way to a late stage therapy, I think is impactful in the market. And I actually think it's a unique way to deliver shareholder return. So it allows us to do that. But we are an investment trust, and the whole investment trust sector, which your American listeners won't know, was also in a bear market at the same time. So we're all trading at a discount and trying to solve that. The future for us looks like where we started, which is let's look at these novel ways to target, to get to novel targets that we can address with all the modalities that are now out there. And I'll give you one example of that, which is we started a company, Yellowstone, out of the University of Oxford. So there's a clinician up there who's a world leader in acute myeloid leukemia. He has his patients' blood samples pre and post stem cell transplant banked for the last 10 years. And within that data set, he has got 15 patients who've got a complete cure with no graft versus host disease. So if you know that disease, you know that if you don't get graft versus host disease, it means the stem cell transplant didn't work because it's not found anything to graft onto. So in those unique patients, clearly there was something on the myeloid cells that was found that wasn't found on a healthy cell. And we sequenced all of those. And what we found is almost all of us, there's 24 novel targets that haven't been described before. Almost all of those are a single SNP difference between donor and patient. And almost all of those are presented on class 2. And that to go to the plausibility of that, so go to where we started, that is a holistic experiment done in a human. That's the best thing about it. So a holistic experiment done in a human where we didn't go in saying it's this target or that target. In fact, look at all targets. What's the result of the model? The result is an alive human being. That's the greatest result you can get. So the way the experiment's set up is just high veracity. Then you go to plausibility. Well, guess what? One of the escape mechanisms for AML to eventually kill you is downregulation of class 2. So it's very, very, very impactful, I think, as an experiment. So we got very excited by that. If you also think about the experimental design, we found these 24 targets because they're effectively the tip of the iceberg with no background noise. They're above the water surface because there's no graft versus host disease. But you now know that if you did a machine learning experiment on that whole data set, looking for single SNP differences between donor and patient, and you ran the algorithm as what likely to be presented on class 2, you're going to find a whole heap more. And so that's a really exciting opportunity, we think, that's just broken up a whole new, not just single target, areas of target and complete over the country. And then you go to modality. How do you address that? Well, you can do that with a T-cell engager. There's a few ways to do that, but those T-cell engagers seem a great way to go after it. And so I use that as an example of the sorts of things we look for and the sorts of places that our future capital is going to go into.
And, I think the very clear question for me from here is, to what degree, you talked about this coming out of a university, but to what degree is this kind of work enabled by the integration of university discovery science with NHS data sets? Because that, of course, is a big part. We're moving to the UK ecosystem part of the conversation now, something that, people in the UK are touting. So to what degree is that part of the linkage?
Yeah, so I think that's very real. As you know, we do have universities embedded into hospital systems. Those hospital systems are NHS hospitals. And so they do have access to large data sets. And that integration, I think, in this new world is going to be very important. And I think gives the UK its place in the industry effectively. So do I think the UK can do what China's doing right now, which is engineer something to the nth degree against a known target? It can do that, but not as fast. And hopefully we get as fast. And I certainly think there's initiatives in the UK to speed up our whole clinical translation. But with 4 of the top 10 life science universities and that NHS system and a relatively big ecosystem outside of the US, we have the ingredients here to do something unique in the industry. And we certainly are focused on that. And I hope other VCs in the UK environment are focused on that because I see it as our advantage.
So, I want to end by sort of going down that road a little bit more. For BioEquity Europe, our European conference next week, our scene setter analysis has looked at clinical trials from clinicaltrials.gov in the last, from 2021 to 2025. And, we really focus mostly on phase 1 in it. We do have a little bit of all of it. And we, people in the UK might be delighted to hear, there's certainly a tier where we're looking at what clinical trials are done where, the US and China are way ahead. But actually the UK really sits in a tier with South Korea, Australia, Spain for some indications. And so, I think about this both in the UK and across Europe, where you do have really, really strong science, which we talk about a lot, but we don't talk a huge amount about the hospital strength, the strength for running clinical trials, and the idea of what I think of is creating a flywheel between the academic discovery and the ability of hospitals to do what you want all the time, which is give me human data, And, so I'm wondering to what degree you think that will, historically, I guess, the physician scientist was much more a part of discovery, like back in the sort of 60s and earlier kind of thing. But to what degree do you think we can really move back towards that closer flywheel from one to the other?
Yeah, I think a lot of the people that are stakeholders in the UK ecosystem see that and are very aligned by behind what you just outlined. So I think there's a lot of effort. I mean, you had an interview with the CEO of the MHRA.
Lawrence Talon, right?
Yeah, Lawrence had a lot of those same messages, I think. And so I think we can move towards that. I think we have certainly got examples in our portfolio where effectively the engagement with the MHRA has offered a quicker route to clinic than we could get in the US. Ultimately, you need to go and do your development in the US because the US is such a big market. It doesn't mean you need to do phase 1, 2. And I think that was really impactful to the success of that company because it meant it could leapfrog its US competition. And given that we do have a very sophisticated and accessible regulator, that's a key advantage as well. And then you get through that and you get to those physician scientists. And the advantage of a physician scientist is that they're researching the area they're researching because they're so desperate for something to treat the patients they're seeing in the clinic. And by definition, that means they know where the patients are as well. And so you have all that connectivity. And I certainly remember some examples of, in our phase 1, 2 trials, our physician scientist founder walking down to the pharmacy himself and getting the material to go back up to surgery. So you do get that connectivity, and I it is another UK advantage, and we absolutely need to leverage it.
Yeah. Well, this has been a great conversation, Chris. We could probably go on for a while, but we have to come to an end. Thank you very much for joining us on the BioCentury Show, and look forward to next time.
Super. Thanks, Simone. Really appreciate the opportunity.
Brought to you by BioCentury Grand Rounds. Join BioCentury and Regional Host Chair Allen Institute this June in Seattle for an R&D forum at the interface of industry and academia.
Kendall Square Orchestra provides the music for the BioCentury Show. The group connects science and technology professionals and other members of the greater Boston community to collaborate, innovate, and inspire through music while supporting causes related to healthcare and education.